I guess after all this time, a few more minutes waiting for this defense, not going to hurt anybody, right? So all of you who've been with this project for a while know that this has taken its time. And there's a great saying in German, "Gute Dinge braucht Zeit," and it means that good things take time. And I think this is an example of that. Brett has been here for, we're not going to mention how long. It's been a long time. It feels even longer because he started life here as an undergrad in our program and did very well there, started getting involved in this research as an undergrad. So it was a long-standing relationship between him and these projects. He made the mistake, and this is the warning to all you PhD students, you get involved helping other people too much. So be selfish, for heaven's sakes, if you actually want to get your PhD done in a reasonable time, and stay focused, of course. So I'm not going to delay any longer. Brett will tell you all about his research, and I hope you'll find it as enthralling and incredibly positive. I have to say the results have been really extraordinary in the end, so I think it has been something that was worth waiting for. So, Brett.
Okay. Thank you, Rob, for that introduction, and thank you everyone for coming here, especially the members of my committee who are here with us today. I'd also like to thank all of the people who've been involved in this research. You'll hear during my presentation that I use the phrase we and our a lot, even though a lot of this is the work that I did, but really a lot of this work is what we did together. And so don't be tripped up on that. I thank you all for being here, and I thank you all for participating and being a part of my research. So to start my presentation, I want to first ask a question. And I want to ask you when was the last time that you were lied to? Can you remember that time? And I don't want a little white lie. I want something that's big, like a really big lie, something where the stakes are high. Like that, that's perfect. No, but I want something where the stakes are even higher, like where your belief or disbelief in what I've just told you is a matter of life and death. I sure hope that this wasn't the case with when we started. I personally have never had an experience like that I'm aware of. And I'm sure that some of you didn't, but there was somebody who did. Tim Russert, who is the host of Meet the Press, who is one of the most influential men of the 20th century, was told one such lie. He was told, first off, I need to kind of give you a little bit of background on him. He was in pretty good shape. He took his statins regularly. He had blood pressure medication that he took regularly, but he exercised every single day. And he even passed a stress test in April of 2008. In June of 2008, he suffered a major coronary that took his life. And that immediately prompted questions that came from various different people, such as, how could this television host have passed a stress test and then 2 months later suffer this major coronary? And these questions came also along with criticisms of our current clinical testing, calling them tests that lie. Now the truth is that all too often our simulation, our clinical approaches can't detect the types of heart attacks that left Luke Russert, Tim's son, fatherless. And I'd like to know why not. Why can't we detect these types of heart attacks? What is it that we would have needed to know in order to keep Tim Russert alive? And is there anything that we can now learn and that we can now apply that can prevent such losses in the future? Well, in order to understand a little bit of how we might be able to go through this, we need to first understand the types of testing protocols that we have. So Tim Russert underwent a coronary stress test. This is a test that attempts to identify some sort of ischemic risk in a patient by increasing their heart rate, either through exercise or some medication that's delivered. And by doing this, they're looking for changes in the ECG that are indicative of an ischemic event, of an occluded coronary or something like that. Now, these tests aren't perfect. Their accuracy is as low as 68%, their sensitivity being as low as 50%, and specificity being even as low as 69%. That means, more or less, if you're diagnosed using a stress test with ischemia, your actual chances of having ischemia might be as low as 50-50. You might be able to flip a coin and tell your doctor how confident you are on that result. Now, specificity, if you're told that you don't have ischemia, you have much better odds. You have a 69% low end, but there's still a 10 to 30% chance that you're still going to have ischemia, even though you passed your stress test. So, but before I move on with the rest of this, this talk with regard to stress testing and with regard to how we can improve it, I need to take care of some housekeeping items. So this project attempts to translate experimental information into a computational domain. I've tried to separate those as much as possible by putting general discussion and physiological information under a blue banner. Under a red banner, you're going to see technical information, things that involve the computational modeling approach. So now that we've got that out of the way, let's talk a little bit about physiology, some basic physiology. What it was that killed Tim Russert was a heart attack. And you can think of a heart attack more or less as a plumbing problem or a supply-demand problem here. Your heart is basically a unit that has pipes going through it, right? And if the pipes get clogged, then you're going to have a supply problem downstream. So first off, your heart starts out relatively open, and your blood can flow through freely. Over time, you might get some occlusion that builds up, some plaques that build up on the wall and slowly start to constrict and reduce the supply that comes into your heart. And if things go prolonged too much, or if you get a blood clot or some other means, then there's a way that that can get completely occluded, in which case you're going to have a heart attack. Now, the ability to detect heart attacks using ECG-based methods is relatively well established. But we're not interested in detecting myocardial infarction. We're not interested in detecting heart attacks. We want to catch the precursor. We want to catch it while it's still in this phase, where it's partially occluded. In this phase, cells still get some blood, but they don't get enough to support their complete metabolic needs. And they start to weaken. They don't necessarily die like you'd have in a heart attack, but they start to weaken. Clinically, it's assumed that, that these injured tissues, these regions that have less blood perfusion, develop more often in the subendocardial domain. That is the domain that is along the blood pool geometry here. It's represented by that orange ischemic region. And under prolonged ischemic stress, it's thought that these ischemic regions progress transmurally, eventually becoming fully transmural. Now, this is a case of transmural ischemia. Anything else that's not this, that's less than fully transmural, is considered a partial thickness ischemia, and that's a term that we're going to be using throughout this presentation. So what we actually see, so this is what the current clinical dogma is. It's assumed that you have these subendocardial regions, but that's not actually what we see through experimentation. Using both canine and porcine models, we were able to find that ischemia develops more as disparate, discontiguous regions that develop and expand independently of each other until they merge together and become a fully transmural zone like this. So we see this, not this, more often than not. Now, throughout the rest of this talk, we'll be referring to subject-specific zones. And the ones that follow the current clinical dogma will either say that they follow the current clinical dogma, or we'll refer to them as subendocardial or some other term similar to that. But what we really want to know is, does this matter? Does it matter if we have a zone that's represented by a subject-specific geometry, or if we can do some sort of simplified geometry like this that always expands from the endocardium? Oops. Okay. And we do want to see if these sort of models would be able to allow us to see errors, or to define errors that we see both in ECG or not. But animal experimentation, which is what we use to gather these, is not capable of showing us what these effects would cause on the human ECG. Nor is it practical or even ethical to do these sorts of simulations or these sorts of studies systematically within humans themselves. Computational models, however, are able and have been employed multiple times to be able to overcome these constraints, these experimental and clinical constraints. And they do have a robust history in cardiac simulation. What you're looking at here is a representation, more or less, of my work, a summary of my work. In it, you can see more or less the technical details, creating a mesh similar to this, as well as the simulation details of projecting ischemic regions onto the body surface. So in order to do this sort of simulation approach, first we had to establish our framework. We had to define something both in the heart that defined the properly defined ischemia within the heart, and also projected it onto the body surface. Once we on the cardiac level, we solved the epicardial potentials based solely off of intramurally recorded ischemic measurements that we took during experimentation. And we also attempted to do a zone-based simulation approach. And you can see a zone in black right there inside of that heart as well. After we'd established that our cardiac simulation was accurate, we transitioned that into a full torso-based approach, and we translated this into into an approach that would be more relevant in a clinical sense. And what we found is potentially paradigm shifting. So we found that the subject-specific zones in some cases yield very different simulated results than the subendocardial or clinical dogma. But in order, before we can talk about our conclusions, we have to explain how we got there. So let's return back to what we originally proposed to do, which was establish our simulation framework. The general framework, or the general purpose of the general framework of our approach has been the theme of the Center for Integrative Biomedical Computing, or the CIBC, that's here within the Ski Institute since it was originally conceived. The point of the mission statement of the CIBC is to develop open source software that can be used for image-based modeling, for simulation and estimation, and for biomedical visualization. An instantiation of that pipeline is shown here, where you can take image data that you acquire from either experiments or some other means, use that to identify specific structures, important structures in your model, convert those structures into geometric surfaces or volumes in which you can perform some simulation. If you have measured information that you can validate with, you can also include this verification phase, and you can loop around and optimize your simulation approaches. Now, this is one example and one sort of pipeline of how things can be done, but I'm going to show you in this presentation how we actually do that in practice. Specifically, for our simulations, we generated this model. Now, don't get overwhelmed by how big it is or try to analyze exactly what's going on. We're going to go through this step by step along the way. One thing that I do want to point out, though, is we do have a little place marker, so you can see where we are. You probably can't read it, but that's okay. You get the general shape, and we're going to follow along with that. In the event that we have a software tool that we use, that'll appear in the opposite corner. So we're going to start this description by talking about something that's not necessarily associated with computational modeling all that often, and that's experimentation. Experimentation informed our simulation approach and was integral to our ultimate findings. Experiments that we did were performed in open chest, porcine, and canine models in which we induced cardiac ischemia, and has been covered more thoroughly by my predecessors who are listed here as references. Now, the overall process is relatively straightforward. What we did is in open chest animals, we fitted the heart with an electrode sock. Now, this is a physical sock that you actually fit over top of the heart that has electrodes sewn into it so that these electrodes can come into contact with the epicardial surface. And those electrodes, represented here as orange nodes, record those potentials that you can see from the epicardial surface. We also inserted plunge needles into the tissues, represented by the yellow nodes. And these yellow nodes basically represent where we took a physical needle with 10 electrodes along the shaft and put it into the cardiac tissue to be able to record intramural potential value changes. In order to induce ischemia, we occluded the LAD, the left anterior descending artery, shown here as this red vessel, with a hydraulic occluder shown here in white. That hydraulic occluder could be inflated or deflated in order to offer different levels of ischemic stress. And once we did inflate those things, and once we did occlude that LAD, we would be able to see electrical changes that could be recorded from these electrode configurations that you see and that have been described previously. But why does a coronary occlusion cause an electrical response? It's not like if we get a clogged drain in our house that our lights dim. But that's kind of what happens in your heart. Cardiac cells are both mechanical and electrical units. That means that they beat, and they also conduct electricity in a very specific way. A healthy cell that has proper perfusion is going to produce an electrical response that looks somewhat like this. This is a cardiac action potential, a healthy one. Now, over the course of a single heartbeat, the billions of cells that make up your heart are going to have a similar action potential to this, and but they're going to be a little time offset. And so that time offset, the summation of all of those cells together with these action potentials will produce an ECG, which is what we're used to looking at. And I wanted to draw your attention to 2 points on these two diagrams. This portion here and this portion here, because under ischemic conditions, they change. Once ischemia is induced, the cells in those regions begin to weaken. They try to get a full action potential, but they can't. And so what happens is you end up getting something that's similar to this, where your lights more or less dim. The amplitude of that action potential just can't quite reach the level that it used to be at. And as a result, you get a potential difference, a voltage difference between points that are ischemic and points that are healthy. These potential differences drive currents, and those currents are detected from your ECG, and you'll see, I'm not sure if you saw this before, so we're going to go back and just kind of go through it. The healthy case, this is relatively zero. This is an isopotential. And so when we do this, when we see an ischemic episode, you'll see that this changes. In this case, it's depressed. Other times it could be elevated. In the case of an ST depression, clinically that's thought to be a subendocardial region, like the ones that we saw earlier. So the metric that we use when we're analyzing these ST potentials, we can't analyze the entire thing unless we want to do a very dynamic model, but so we take a single time step where we want to see how that ST segment deviates. Does it go down? Does it go up? And so we extracted a location that is 40% in between the QRS spike here and the peak of the T wave here. So 40% between those is about right here, and we call that ST40%, or just more simply ST40. It's also important to note that these ST40% aren't only on the body surface. These can also be measured. You can measure potential differences, drops in potential or elevations in potential within the heart through the needle electrodes as well as on the epicardial surface. So let's continue on with our simulation pipeline. We've covered experimentation, and after experimentation, these signals from all of these electrodes were extracted and processed using the Pfeiffer signal processing application that we have here at the Ski Institute. And ST40 potential values were extracted using this. We also extracted volumetric data using MRI and fiber orientation data using diffusion-weighted MRI imaging that we subsequently mapped into our mesh, and I'll explain that when we get to that phase. So up to this point, we've covered 3 components of our pipeline that aren't necessarily as related as we think to a computational framework, but they're all very integral in being able to process the next 8 steps of our simulation pipeline. We'll begin this part of the simulation pipeline by first considering what we do with our imaging data. Well, first we take that imaging data and we segment it using the SEG-3D software package that's developed here at the CIBC to create subject-specific volumetric data. That volumetric data is then piped into the Biomesh 3D software package in order to generate high-resolution, surface-conforming meshes of both cardiac tissue and blood geometry. Now, the sock and the needle data that we collected from our experiments is something that we want to impose into these meshes, but it's not all automatically in the space that it needs to be in order to do that. And so we need to go through a registration process, and we use the SkiRun software to be able to register this information within the volume. This isn't a trivial step, though, however, because we have 2 domains. We have a sock domain, we have a needle domain. Actually, we have 3, and we have a mesh domain. And we need to get everything all in the same spot and communicating well. And so I'm going to take a little bit of time to kind of explain how we do this. The first part of this phase, to explain the first part of this phase, we have to go all the way back to experimentation. After our experiments were over, we digitized specific points on the sock, again, remember the orange electrodes here, and the needles, the yellow electrodes here. And you can picture it like we did this. So we took the needle electrodes, those are our green points. We took corresponding nearby sock electrodes, those are your magentas, and then we defined them as saying that these 2 corresponded, which means that if I know a sock electrode location, I more or less can know where the needle is that corresponds to it. And we used that to map the information into our mesh domain. First, by identifying needle locations. See, needle locations were available in our MRI data, so that part was easy. We've got the needle locations now, and now we need to get the sock into that space. So how do we do that? Well, one of the things that you also need to notice here is that these yellow nodes, these needle electrodes, they're all more or less on the frontal plane, right? They're in a 2D space. And that's a really hard thing to register a 3-dimensional object into a 2D space. And so what we needed to do is, in order to overcome this, is we took a sock template geometry. And because we knew the corresponding nodes on the sock that corresponded to the locations in this mesh space, we were able to do a rigid transformation, keep that geometry of that 3-dimensional space, and just transform everything into that mesh space like this. Once we had this in place, then we needed to turn to other physiological measures to get better correspondence points, because we don't have correspondence on the back, and there's potential that we could get better correspondence once we're in this space. And so what we did is we turned to activation times. we assumed that if these electrodes are close by, they'll have very similar activation times. But what is an activation time? You can think of that in terms of a ripple. When you throw a pebble into a pond and you get a ripple, once that pebble hits the pond, you have an activation point, a site of activation, and then the ripples emanate out of it. Now we can pick a point somewhere else on this ripple plane, and we can time it. That's basically the activation time of this point on the ripple. If we pick another point that's farther away, we might not get the same sort of activation time. In fact, this would be a very quick activation time. This would be a little bit slower activation time. If we picked a point that was close by, these might have very similar activation times. Now, the heart propagates electricity in a very similar way to a ripple, as a three-dimensional wave through a volume. And you can see how regions that have close points that are close together, you'd have a similar activation time. I do have to acknowledge that there are instances where you would get a very close activation time, but it wouldn't be a good correspondence point, as right here. So you can see that right here you have an activation time that's going to match the activation time right there, but they're not in the same local neighborhood, which is why we did that first registration. We could constrain our choice of correspondence points by the local neighborhood that we already established by getting that first rigid transformation. And so now with things in place, we identify the activation times along the entire epicardial sock, and we also identify them on the epicardial most needle point, which is about on the epicardium. And we used those values to create correspondence between these two domains and snap our mesh into a much better configuration. So with the mesh in the configuration, with the needles in place, we could then map information into the mesh that we had. In the event that we had a zone-based simulation approach, which wasn't all of our simulation approaches, but certainly some, we used zone-based approaches, we would map that information into the intramyocardium, and then we would apply a threshold to be able to extract the, oh, that didn't work out, to extract the zone. What you would see here is there's a thresholded zone in here that's surrounded by some other tissues. And that we ended up using to pipe back into the Biomesh 3D software package to create conforming meshes that had tissue, blood pool, and ischemic zone geometries. With all of that information in place, we now need to define conductivity within our conductive medium, because the heart is a conductive medium, otherwise it couldn't pass electricity. To do this, we used diffusion-weighted MRI to extract fiber orientation. Now I need to kind of step back. The heart is a fibrous structure. It has muscle tissue that is elongated and that conducts preferentially along a certain direction. This is known as anisotropic conductivity. So it would rather go down along the fiber orientation of your cardiac muscle than it would want to go across it. And so we need to define that. And the way that you do that is you use diffusion-weighted MRI, and you figure out the principal direction of flow. And with that principal direction of flow, you can identify a vector field that you can map into your mesh. And then you can assign conductivities to that vector field, allowing the electrical conduction to flow preferentially along one direction, but you're in a three-dimensional space, so it can technically flow in any direction that it wants. But you can constrain it basically with your conductivity parameters, saying, no, we want you to flow preferentially in this direction rather than this direction. So you set it up as more or less a tensor field of conductivities. All right, now we have each component of our model in place and we're ready to do a simulation. We computed extracellular potentials first using intramyocardially recorded information from experimental models. What you're seeing in this image is a three-dimensional geometry of the heart that has embedded inside of it the needle positions. The coloring on those needle positions represents the potential field that those needles registered. So we took this information, we mapped it into our mesh, and we defined the epicardial surface based off of that information. The way that we did these simulations depended on our boundary conditions and our source configurations. In this particular case, we assumed that we were only in the extracellular space. So we could solve this assuming that this was just a volume conductor using Laplace's equation and a finite element method. Our second cardiac simulation required zones. In the zone-based model, we defined not only the geometry of the zone, but we also assigned a source, which we defined to be the transmembrane potential between healthy and ischemic, the transmembrane potential difference between healthy and ischemic tissues. We used what's known as a passive bidomain to solve this equation at a single point in time using a finite element method. And a third simulation approach that we did apply comes in a little bit later because it's a torso model. What we did here is we extracted the information that we pulled from our second model here, the solution of our zone-based approach, and we mapped that into a torso phantom. Using that torso phantom, we assigned a conductivity, an isotropic conductivity value with which we could define solve for the body surface potentials. And then once all of that information was in place, we made our solution. It's now time to validate because we do have this information. We extracted needle data, which provided our source, and we extracted epicardial sock data, which provided our validation point. I don't want you to analyze this now, but we're going to look at this a little bit later. But this is the types of results that we can see. And you can see that there is quite a bit of correlation between simulated potentials and measured potentials. And we'll talk a little bit more about how correlated those are. So there you have it. That is our complete ischemia simulation pipeline in its entirety, the construction of which was highly motivated by errors that we observed in ECG-based diagnosis, as well as observations that we made in experimentation, where we saw ischemic regions form that weren't explained in a clinical sense. But so now that we've got that simulation, that pipeline put up, let's try and bring this back to people. How can this help people? Because that's really the motivation that started this whole thing. Is there a way that we can improve our clinical diagnostic techniques in such a way that we can prevent this loss of life that we've seen in the past from either failed misdiagnosis, or we can prevent poor costs in our hospitals by admitting patients that don't need to be admitted. And the way that we propose to do that is through simulation. Yeah, I believe that we can improve our diagnostic techniques, but it's going to take some reworking of our current thinking. And we can rework that through simulation. So first we're going to talk about each simulation in detail, starting with our intramural, replicating the epicardial potentials based on intramural measured extracellular potentials. And this work is in its final review phase at the Annals of Biomedical Engineering, and will soon be published. I haven't got word on that yet, but it's very, very close. Before I begin with how we describe this process, I want to re-emphasize that experiments really informed these models. And our experiments showed that instead of the current clinical dogma, we see ischemic regions that don't conform to that idea that come in the mid-myocardial space or in the epicardial space. These experiments, in essence, broke the clinical dogma. It broke the theory that everything starts on the subendocardial space and expands transmurally. Instead of seeing subendocardial regions, we saw regions more like this. And we therefore needed a new way of representing ischemia that was not only more physiologically accurate, but could still produce the information that we needed on the epicardial surface that was even more accurate than our current clinical thinking. The first step in this process, as we've already described, was to build out was to map in the measured potentials, which we could then use to simulate a forward solution to the epicardial surface, which we could then validate. Now what you're going to see here is our first visual attempt at validation, right? So you're seeing two different orientations of the heart right now. You're seeing the frontal plane, the anterior view, and you're seeing the inferior view, which is basically like turning the heart and looking at it from apex to base, right? From tip to top. So what you're seeing in this, there's some things that are immediately apparent. You have ST elevations that appear here in the apical region, which is downstream from the occlusion that we placed about right here. So you get these elevations in that apical region. You get depressions that flank that area. But one of the things that and you see that also on the measured potentials, which is very nice. But one of the things that you don't see, which isn't very nice, is this depression here. This is our measured potentials. Okay, in measured potentials, you see a depression here, but you don't see a depression here. Now, the thing that makes this a little bit more clear as to why we don't see that is we can come over here to this clipped needle hole region. Now, what the clipped needle hole represents is the area that's just over the needle region. Remember how you saw those yellow nodes? This basically takes the boundary of those yellow nodes, because we want to see exactly that complete underlying ischemic region. But if you look at this and you look at the inferior view, you notice that even the experimental stuff doesn't see it here, the experimental measured information. So the answer to why we don't see it here is we just couldn't sample it. And there are a lot of restrictions in experimental-based models. The lungs get in the way, where your incision is, your pericardial sock as you're trying to analyze these. There are reasons that we couldn't get the sock into that space, but without that information, we can't replicate that sort of signal. We'll take a look at another example. This is the same sort of example, different heart, different animal. You see elevations, multiple elevations overlying the ischemic region. You see a depression that flanks it on the basal side, and there's actually a depression over here on the left ventricular free wall that we see in both simulation and measured potentials. But again, you don't see that inferioapical depression, that depression right here along the apex. So though these simulations do produce quite a bit of agreement, they're not perfect. There's room for improvement. But they do produce relatively high correlation between measured and simulated approaches, which can be shown here. What you're looking at here are the Pearson's correlation coefficients for every single heart that we ran. We ran 226 of these, 226 different ischemic interventions, 226 simulations, and we compared them all. And we then noticed a pattern here that if we defined our ischemic stress in terms of the maximum epicardial potential, if we found experiments that generated 3.5 millivolts or higher, we actually could predict that our simulation approaches were going to be relatively well correlated. There's only one example where this didn't happen, and that's this orange case here. Now this belongs to a specific experimental subset. And this experimental subset, for some reason, just didn't come together just right. The needles actually looked perfect. They looked like every other needle set that we had, but the sock apparently seemed to be dissociated. And we try and explain that away using either maybe there was something that happened experimentally. One of the theories is that we allowed the sock to dry out, right? So that sock needs to stay moist so that you get a good connection on the epicardial surface. And maybe we let it dry out and it became disassociated. Maybe it even slipped off. But either way, that one experimental subset just didn't quite work out. Still, we were able to see that if we had 3.5 millivolts recorded from the epicardial surface, we would get 75% of those that are above that threshold to have strong to moderate correlation. If we removed that orange space, then we could improve that to 83%. And over the clipped needle hole, we have just very high correlation as well. An easy way to kind of visualize that is using these sort of plots, where you have, if you have a high ischemic stress, you can see that you're going to have, generally speaking, a strong correlation over both the clipped needle hole and the complete epicardium, and you're going to have much less variability. So what we've done so far with this is we've simulated the epicardial potentials based solely on measured intramyocardial recordings. With this, we were able to verify that our simulation pipeline, our simulation framework, is actually relatively accurate in reproducing the epicardial values that we see experimentally. This gives us hope that this framework would work would be something that we could use moving forward to help us improve our ability to better detect clinical instances of ischemia. But a doctor's not going to look at somebody's ECG and think to themselves, well, I bet there's a point of elevation here and a point of depression over there and a point here, 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 and here. They're going to look at it and they're going to try and think of it in terms that are much more feasible. That's impracticable for anybody to do, is to think of it on a point base. What they do is they think about it more or less in terms of ischemic zones, which we've already shown. Their current model is a subendocardial ischemic zone. Or they could think of it in terms of vectors that are created by ischemic zones or things like that. Either way, they're going to think of something in terms that are a lot simpler. And so we proposed to move forward with an ischemic zone-based model. Using our experimental approaches, again, to inform this model, we generated simulations that used three-dimensional geometry pulled from experiments. This is in its first round of reviews at the Journal of Electrocardiology currently. So again, similar to the first attempt that we made, we used these zone-based models as opposed to the intramyocardial models. The needle-based models is what we'll call them moving forward. There's zone-based, needle-based. We use these zone-based models to compute the epicardial potentials, to simulate them. From those, we again tried to validate them against measured epicardial recordings. And we saw if there was any sort of improvement. Now, zone-based models differ from the previous needle-based models in that they don't consider every point that we took. They look at all of the points and then they say, I want the ones that are the highest potential. I want to threshold out the ones that have the most influence. And so they create an ischemic zone. But these ischemic zone-based models also have some idiosyncrasies about them. because let's go back and look at that original image. This isn't a binary image of potentials within this field. You don't have something that says this is on and this is off, this is ischemia, this is not. You actually have a graded field. And so when you do create that, that threshold that you use to define your ischemic zone, you also need to consider the regions outside of that threshold, how they still might influence your solutions. In this way, the concept of the border zone is created. Now, the border zone is a region of moderately ischemic tissues. It's not ischemic enough to generate a full threshold to say, yes, you're an ischemic region, but it does have some influence. And it looks like my next slide is going to have another blacked out image. This is the same image. In the image, what you would have seen, I don't have to see a marker, but okay, what you would have seen is you'd see an ischemic region, and in three-dimensional space, you'd see the shells that overlaid that ischemic region. And it more or less shows you that with your ischemic region, you can still have some influence on the outside. We chose to define our border zone using, and that's where it would have been. We chose to define our border zone using an approach that was previously proposed, in which you have a smooth transition from your original definition of your border zone down into a region where you then transition into a sharp transition phase. And this was found to be necessary because if you don't have that sharp transition phase, you actually don't get the depressions that we see. The way that it was defined is by using a Gaussian decay function at the very beginning, and then transitioning that into just a linear function that then goes to 0. And what we're interested in is we're defining healthy tissue as having a 0 potential. We're defining our ischemic tissue as having some threshold potential, which in this case, in this diagram, is 30 millivolts. But every single one of the parameters that you see as part of this definition is something that we can manipulate to improve our simulated solutions. And we did. And we found that if we manipulated the transmembrane potential, that is the value of your y term here, you could essentially scale your epicardial solution. If you increase the transmembrane potential, you'd increase the range that you saw on your epicardial surface. If you change that Gaussian variance, you would essentially translate your epicardial potential. So if you increased that variance, you would actually decrease overall. You get a more negative value in epicardial potential. Now the S1, your edges of your border zone, those were more or less used to fine-tune things. So you could actually get these epicardial surfaces that shared more or less what you saw experimentally. You could fine-tune your simulation approach to basically match, at least as far as epicardial potential is what you saw experimentally. Now this S1 term, it looks like it doesn't have anything going on, and it doesn't change your epicardial potentials. But what it does do is it creates that blurring region, how long it is, and it actually increases your correlation as you change that without changing your epicardial surface potentials. So what we're going to look at here are a set of different experiments that we did using zone-based approaches. And these are all done from 2 different experiments. A1 through C1 is experiment 1. A2 through C2 is experiment number 2. And these progress in time. So A1 was performed before B1, which was performed before C1, and so on. What you can see with these simulations, we did produce relatively correlated, more or less, potentials along the epicardial surface that measured up with the measured potentials in this way. Now, as we go in time from A to B to C, you can see that there's a little bit of a dissociation. It appears that there's some dissociation. That over time, things seem to get a little bit worse. But actually, what's happening is that's a scaling issue. Based on Pearson's correlation coefficient, these are actually still strongly correlated. They might not look like it, but the distribution of potentials on that surface is still very correlated to what you see in our simulated approaches. Your errors do increase, but your correlation doesn't decrease much. One thing that I do want to show that's very important, I'm going to show it here in this A1 phase, is something that we didn't see in our needle-based approach. So in our needle-based approach, we were able to see these depressions on the lateral free wall. And if you recall, we weren't able to see inferioapical depressions. However, when we introduced zone-based models, they did produce inferioapical depressions that more or less lined up right where they needed to with our simulated approach, with the measured approaches, which gives us hope. The introduction of a zone allows us to reintroduce data that we lost along the way, which is really nice. And I could explain why that is, but that's probably something if you want to ask, you can ask me later. So right now what I'm going to show you is a lot of data. And in that data, we're going to do a comparison of our zone-based approach to our needle-based approach. And I'm going to try and pare it down so that it's not so overwhelming. But at first, it's going to seem a little bit, oh, things are gone. There. All right. So what we're going to do is we're first going to pare down all those numbers. And we're only going to look at Pearson's correlation coefficient. You can see that in the cases of our zone-based models, which are here, the Pearson's correlation coefficient surpassed that of the needle-based approach every time, and in many cases by quite a bit. Now, if we look at a different metric, we're going to look at what we call absolute error percentage. And this is a metric in which we looked at what percentage of the epicardial surface had an error of greater than 1 millivolt, which isn't a huge error, but we wanted to see how much had an error greater than 1 millivolt. And you can see that in many cases, the zone-based approach still did better, and if it did better, it did a lot better, like as you see in these cases, but it wasn't consistent. So it did show that there was some improvement, but it doesn't necessarily mean that it's a consistent improvement. So what we were able to do is we were able to use zone-based models to simulate epicardial potentials. And those epicardial potentials that we simulated were correlated to measured potentials, but they were also more correlated. They seemed to agree more with those measured potentials than our original needle-based approaches. We also discovered the influence of different parameters in our border zone on epicardial potentials. Now this allows us to say that we can transition this information into a zone-based approach. And remember, the zone-based approach is going to be much more transferable to the clinic. They're not going to think about points. They're going to think about something that's easy to visualize. A zone is somewhat easy to visualize. But something that would make even more clinical relevance is if we could take this to the body surface, which is what we proceeded to do next, by imposing these values that we solved for into a torso phantom and solving for the body surface potentials. So in order to do this, we had to, again, do another registration approach. We had to register the heart into the torso. We did this first by taking a known skeletal architecture and imposing our torso phantom over top of that architecture. Once that torso phantom was in place, we could position the heart in the spot that it needed to be, based on also the data that we got from this torso model. Also, we could use this information to position the 12-lead ECG and make it so that we could extract the ST40 potentials that are important for clinicians. So we're going to consider a few examples here, and we're going to consider them against the current clinical dogma. We have our subject-specific zones, which we define in our simulations, which we define with anisotropy and with border zones and things like that we've determined or that have been determined to be accurate and necessary. And we're going to compare it to the current clinical dogma, which actually assumes isotropic conductivity, no border zone. And we're going to see what happens as they compare. So first off, you're going to see images that have these markers so that you know exactly what you're looking at. And we're going to show what they did to the body surface potentials. And then we're going to show you as well what that means on your ECG traces. So on these, you'll also see that there's these red dashed lines and these blue dashed lines. These are the threshold values in the clinic. If you have 2 consecutive electrodes in your ECG that are above that threshold, then you're considered, the red threshold, you're considered to have fully transmural ischemia. If you have 2 consecutive electrodes that are below the blue line, you're considered to have subendocardial ischemia. That's kind of the brute force metric that they use. So let's first consider what happens when we look at a subject-specific ischemic region and what it does to the body surface. You can see that you have elevations in V2 through V4, which indicates that you have a transmural zone. You actually don't. You have a mid-myocardial zone, but I'd rather be told tell a doctor that I've got something that's elevated like this so they can fix me quick, rather than show a depression, in which they might try and use medication first. Now, if we compare that to the subendocardial zone, like this one, you can see that the body surface potential maps are very different. We tried to make them somewhat equivalent in volume. They're not completely perfect, but what you see is you have an elevation over the ischemic region here, you have a complete depression over the ischemic region here. In fact, all 6 of your precordial leads are depressed. This is a huge difference that is not being taken into consideration. Next, we're going to consider a completely transmural case with isotropic conditions, no border zone like you would consider in the current clinical theory, the current clinical thinking. And what you see is you do have a similar pattern, you have an elevation at least, but when you look at those ECG traces, you're actually registering, yet again, an ST depression in V1, V2, and possibly up to V6 even, just not super severe. Now, if we add anisotropy, things change. Just with the inclusion of anisotropy, you're going to get potential values that much more reflect what you would see normally. So we already know that in our modeling approaches, it's already been argued that anisotropy is important. This makes a case that it's really important if we want to see what's going on. Now, if we go the other direction, we take our subject-specific zone, we apply clinical theory, where it's isotropic, no border zone, and what do you expect to get? Well, clinical theory assumes that once you have a mid-myocardial zone, you have silent ischemia. This obviously isn't the case. You should have a high elevation in this case, but the clinical theory does not support that. And so what we've done in this is we've shown that we've transitioned our simulation approach into the torso, and we've shown that by so doing, we're able to create simulations that don't necessarily reflect what is assumed to be true with the current clinical dogma. This is where we see a paradigm shift, where we need to teach that these sorts of things are not what's going on. We need to have a change in the way that we think about how ischemia develops in order to improve our ability to diagnose it, and in order to be able to save the lives of people who might otherwise be fatally misdiagnosed. The ultimate goal of computational simulations like these is to eventually get to a point where our computational simulations can for lack of a better term, beat clinical detection, where we can actually use computational methods, apply it to a patient, and be able to tell them, oh, look, this is where your ischemic region is, this is how severe it is. But we're not quite there yet. This research has been a step forward along that path. But the ultimate goal of these types of simulation approaches is to create something that we call an inverse problem, where you do basically what the physician does with the computer. You take information that you can record from the body surface, and using that information, you can solve for the sources within the heart. That being the case, we first need to develop strong forward simulations that can then be converted into those inverse problems if we want to have any sort of robust ability to turn what we've discovered here with our experimental models, what we've discovered with our computational models, into a method of improving our clinical diagnosis and creating more accurate diagnostic approaches with computation and turn that into a reality. So here are the publications so far that I've had along my journey. They include publications involved with experimentation, with computational forward modeling, with computational inverse modeling, with uncertainty visualization, and of course the topics that we've talked about today. I'd like to acknowledge the members of my committee who are all here, and thank you for all of your help along the way as I've gone through this process, and all of the other people who have helped as well, specifically the members of the Ski Institute, Brian and Wilson, who are over in the lab at CBRTI, Jess, who's here, others who have since left. Is Dan in here? I don't see him. Yeah. But these are the people who helped me here at the Ski Institute. Also the members of the CBRTI who helped with all of our experimentation. The members of the Small Animal Imaging Corps who helped with the visualization approaches. And of course, I wouldn't want to leave out my family who've been with me. Thank you very much, and I'll entertain any questions.